Hello, hope you are good and doing well. Today I'm going to present about the new mixed trends in crop plants under salt stress condition. So it should be based on the phenotypic, physiological, and molecular aspects that we can use and to determine the genes responsible for the salt tolerance under biotic and abiotic stress conditions. So in this video, I am talking specifically about the abiotic stress and specifically the salt tolerance conditions, how we can deal with that and how we can get the salt tolerance genotype and we can use for further functional genomic analysis. So we can use these genes and proteins that can help us to tolerate under the stress line conditions. So I briefly discussed the introductions about the salinity as well as some techniques how we can determine step by step the physiological parts, agronomical parts, that molecular, and in the molecular we can discuss specifically the RNA sequencing, genome-wide studies, and how we can use the genome-wide analysis. So if we talk about the abiotic stress, so abiotic stress, particularly salinity and drought, create a negative impact on plant growth and development with ultimate significant losses in the production of essential food crops. So we can see from the figures here the amount of the food globally that we need, but the projected food that we need with the increasing population is less due to the abiotic and biotic stresses due to the climate changing environment affect the food security specifically. So there is a need to determine such type of genes that can withstand and work very well under specific conditions including salinity. So, salinity is an important factor that limit the growth and crop productivity. Worldwide, salinity affects 20% of the irrigated lands and consider 50% in the future as the condition prevail. So, it should be know about the salinity to know what is the saline condition. So we can say that a soil has electrical conductivity about or above 4 decisiemens per meter considered saline soil, which is equal to 40 millimolar sodium chloride. So if we talk about the effect of the salt stress on the crop plant, so it will affect the seed germination at the first stage, and after that restrict the water uptake in the soils, and after that the inhibits root growth and cell elongations, inhibits leaf growth and new leaves production, leaf senescence, oxidative stress, inhibits protein synthesis, inhibits enzyme activity, and inhibits photosynthesis, and finally and ultimately the yield reduced due to the salt stress. So there are a certain type of plants we call halophytes. Halophytes may use avoidance mechanism and glycophytes tolerate salinity by reducing ion imbalance, and as a result strong secondary effects occurs, and plant adopt certain tolerance mechanism to withstand against salinity in the field condition. So tolerance mechanism require different type of actions, mechanoreceptors, ion transport channels, and secondary signals molecules to maintain ion homeostasis. So when we are facing the salt stress condition, so three types of responses occurs are showed by the plants. First is the physiological response. So the loss of turgor, reduced of leaf water potential, decrease stomatal conductance, reduce internal carbon dioxide, reduce photosynthesis rate, reduced growth, leaf senescence, and biochemical response. In the biochemical response, the osmolyte accumulations, oxidant enzymes, then change in the photosynthesis pathways, induction of plant hormones, and ion homeostasis mechanism are involved that can affect under the control and the saline condition. So number three is the molecular response. The molecular response, the stress responsive genes upregulate or downregulate under certain conditions. So number of genes control salt uptake, gene with osmotic protective functions, genes that control cell and tissue growth rate. So the first target we can say that the screening genotypes based on the different physiological and agromorphological traits could give an idea how we can mm, finalize some genotypes that we can expose under different conditions. So at the end, we have a sensitive, a tolerant, and the resistant genotypes available. And from here, we can further use for the genomic analysis. In the second, we can say that from the first screening, we can use these genotypes and identify the differentially expressed genes of salt tolerant genotypes using the next generation sequencing platform under a specific condition. For that purpose, we will go for RNA-seq and transcriptome analysis under the control as well as the saline stress conditions. And from this data, we can select some salt tolerant genes and we can use these genes and validate the study finally using the qPCR or the real-time PCR analysis. So activation of cascade gene in saline condition trigger hormone breakdown, signal transduction pathway and stress responsive genes available when a crop exposed to the saline conditions. So there is at the end of the RNA sequencing and transcriptome analysis, we have a differentially expressed genes, we have archaic pathways that is changed under the control and the saline conditions. So we can use these pathways and the other gene that is expressed under salt conditions provide the understanding of the mechanism under the stress condition, and it is important for the breeding as well as the functional genomic analysis specifically for a specific genome or for a specific crop that we are studying. So, if we can say that there are a number of different defense mechanisms adopted by the plants against stresses, which are controlled by single or multiple genes in nature. So multiple genes are controlled by a number of transcription factors. So a transcription factor is a specific condition that is used, that is available or in a specific condition. I mean, if plant exposed under high temperature, low temperature, salt stress, drought stress, and the nutrient stress. So these genes are group of genes activated at a certain condition. So these defense mechanisms and their pathways in plants are heavily investigated with genetics, genomics, proteomics, and metabolomics. So these are the new trends in the agriculture and the tools that we can use and get a deep information about a specific crop. So if at the first stage for morphological and agronomical traits, if we can determine and check the response under different saline conditions, say we have the control and we are using different salt stress level 50 millimolar, 100 millimolar, 200 millimolar to check a certain level where a specific crop can withstand a salinity, and after that we can go for the full experiment. And in this full experiment we use a number of parameters we can use to check the response of a certain crops under saline condition. So at the end we can use different uh, statistical analysis techniques to finalize the results. And in the physiological or the biochemical analysis, we determine the total chlorophyll content, stomatal conductance, leaf temperature, relative water content, protein content, total soluble sugars, photosynthetic pigments, sodium and potassium ratio in the shoot and as well as from the root, and their concentration also. So it will give us the idea and the response under the salt stress condition. So for that purpose, if we go for the analysis for the agronomical and morphological character, so we can use the descriptive analysis. It will give an idea or the range between the genotypes, minimum and maximum values, and the mean and mode values, and after that we can determine the ANOVA, the significance level of each level that we are using. Either 50 is near to the control, at the 100 is far away, and how's the response of the plants for each character. And after that, we can go for the correlation analysis, principal component analysis to check the biplot and the distribution of the genotypes were present in first quadrant and the characters that we are using to analyze. And after that, we can, if we have more than 5, 6, or 10 varieties that we are using in our experiment, so we will go for the cluster analysis, making the grouping and determine which type of genotype is making a group with the other one. So from here we can determine which genotype is sensitive and which is the moderate and the tolerant one. And finally we can check the salt tolerance index. From the salt tolerance index we will get the more sensitive as well as the more tolerant genotype under the salinity condition. So in the first stage, we will go identify the germplasm, the genotypes of the cultivar that we have for its sensitivity, moderate, tolerant, and finally the tolerant. So here we can, these three genotypes are, I mean, even one or two genotypes, it will help us to go for the further analysis. So in from here we can determine, we can choose one or two genotypes and use it for RNA sequencing. In the RNA sequencing we will go for the germination of the genotype and under the control and the stress condition at specific level, I mean 2 leaf stage or 3 leaf stage, when the condition, when the plants is adaptive, so we will get the, we will apply the stress, and after 6 hours, 12 hours, 40, 24 hours, and 48 hours, 72 hours, and finally the 96 hours, we can expose the plants for a specific stress. Here we are discussing about the salinity. So we can expose and collect the samples for RNA, for total RNA. And after this total RNA, we will use for the sequencing and transcriptome analysis, because the RNA expression is different in different tissues at different times. So this is the main theme behind why we are using the samples at different times and different tissues. So RNA sequencing basically is a biological measurement convert to the sequencing from DNA and high throughput sequencing. After the throughput sequencing, they will construct the library and the read data, and after that they make the analysis, inference and estimation based on the control as well as the stress, and after that we will go for the estimated measurement. So at the end, finally, we have different transcriptions, translations, and protein-protein interaction, 3D structures, and the variations among the plants under the control as well as the stress condition. And we have different number of expressed genes, differentially expressed genes available at the end of the data. So that's why scientists now focus the RNA sequencing at a specific stage. RNA transcript and their abundance captures the dynamic state of a cell at a given time. So in this case, the DNA making the transcription and making the translation, and a number of proteins are formed under specific condition. Under control condition, the same protein show differently relative the protein formed under stress condition. So during the translation or the RNA sequencing, the transcriptome analysis, it will give us a different results relative to the control. So that's why we are using the biological control in the samples to determine the analysis. So basically this is the advantage or the good things that we are using for RNA sequencing. So different transcripts are present at different abundance. Transcriptome is a mixture of transcripts from all the genes. So here a little bit comparative studies of the Sanger sequencing and microarray based methods. RNA sequencing provide for higher coverage, greater resolution of the dynamic nature of the transcriptome, the discovery of the novel transcripts, identification of alternatively spliced genes, detection of allele specific expression. Now the cost for RNA sequencing is clearly down when if we go for the check the 2001 for one cost per genome is 100 million dollar, but with the time you can see here up to 2019 it's about $1,000. So it provide the facilitation for the researchers as well as the scientists to manage the things easily relative in the past. So here is the main phases that we can use and determine the salt tolerance level. In the first step, evaluate salt tolerance different genotypes using various morpho-agrophysiological traits, screening genotypes against molecular markers related to the salt stress. And the second step or second phase, gene expression profiling of different gene groups, stress perception, stress modulation, and effective uncertainty stress on genotypes differing in their salt tolerance. Gene validation selected from transcriptomic library developed from leaf and the root on the basis of salinity tolerance level that we are using in our studies. So you can use the three different levels including the control. It depends upon the crop that we are using, either it's the salt tolerant or the salt sensitive. So based on that we can select the salinity levels to expose the plants and determine his response. So the samples will be collected dipped in the liquid nitrogen for RNA extraction, and even you can use the triazole methods. Nowadays we are using the RNA laters to save the plant's tissue for better results. So after the total RNA extraction, we will check some sort of quality parameters for the machine. I mean the high sequencing, throughput sequencing, next generation sequencing. At this stage, we can determine the genes related to the signal perceptions, signal transduction, and effectors related to the salt tolerance from the transcriptome library. So for the sampling at the tissues that we can use both at the root level as well as the shoot at the leaf levels. So and exposed for different exposures of time. So that's why I prefer that we can use the 6 hours, 12 hours, 24 hours, 48 hours, 7 to 2, even the 96 hours. The maximum transcript should be available at 6 hours to 24 hours. It will decline or increase based on the plant response under the condition. But relatively control, we have a better response at 6 hours because at this stage, plant should respond against the salinity. And at the end, we see we can get the number of differential expression genes under each specific stage. So based on this data, we make the SSR primers from this transcriptome library developed under the salt stress conditions, and these give a very good indicator for the rest of the family. So we can use these primers for salt tolerant to determine the salt tolerant genotypes. So basically this is the step-by-step procedure that we are using for RNA-seq and transcriptome. So it's a sample collection, sequencing, then cleaning, then quantification. So by this way we can making the inference analysis finally at the end. So the genes that we are using, these genes should be validated using the qPCR. And using the qPCR, we can determine the expression level either is upregulated or downregulated. So we can use any of the stage. I mean, 6 hours exposure to the salt stress, 12 hours and 24 hours, and you can use these selected genes from the transcriptome library. We can use for the validation. So this what delta ct value would making help us to determine the expression level of the genes relative to the control. So for an indigenous control, we can develop our own or sometimes the actin genes used as the internal control or biological control during the qPCR analysis. Our second most important is the genome-wide analysis. So a genome-wide association study is also known as the whole genome association study in an observational study of the genome-wide set of genetic variants in different individuals to see if any variant is associated with the specific trait, with the specific genes, with a specific protein family uh, under a specific conditions. So we can determine all type of data for a specific type of biotic and abiotic stress. So for that purpose, if we go for the advantage of the genome-wide analysis, so biological pathways of the trace does not have to be known, and discovering novel candidate genes, characterization and identification of protein families, transcription factors, specific genes under specific conditions should be available at the end of the analysis. For that purpose, the whole genome wide analysis was done to determine the relationship among the specific protein, gene, and transcriptome family under biotic and abiotic stress condition to understand their function of the specific domain that we are using are studied. So application of RNA sequencing would help us provide the data of the transcriptome and the number of genes expressed and the transcripts available in each specific condition, I mean biotic and abiotic stress. So we have the gene characterization, functional genomic studies, and gene expression analysis at the end. So with the aim of to find the differentially expressed gene and its associated pathways with comprehensive of cropland transcriptome at different growth stages under saline condition can be validated also by the genome-wide analysis study. So for the genome-wide analysis, we use different steps. So from here to upward, I will talk about the steps that are using for the genome-wide analysis. The first step is the gene identification. For the gene identification of a specific protein family or transcriptome family or specific genes working under the saline condition, so we can identify the gene from the genome. From the genome, we should download the data from the NCBI as well as the phytosomes. Uh, Phytosome and the NCBI provide the full genome tabular form. So in this case, you have all the information of all the protein, the accession numbers, the size of the protein, and PI value, the weight of the protein, all the things you can find from the tabular and genome data. And the second step is the conserved protein conformation. In the protein sequence file was used to find conserved protein sequence using the CLC sequence viewer. And after that, the third step is the re-verification of the protein sequence. For that purpose, we are using the BLAST-P program at NCBI web server further verified the putative protein sequence that we are using to determine. Number four, the additional information. The NCBI database was also investigated and marked specific gene information, including the accession numbers, chromosome numbers, genomic details, protein size of each selected sequence. And for that purpose, we are using the program XPACY Bioinformatics System Resource Platform was used to measure molecular weight and isoelectric point. Number fifth step is analysis of the conserved Moth structure. For that purpose, we are using one online tool that is directly linked with the NCBI. We call it meme suite. For the meme suite, we use the protein sequence and to find the conserved motif analysis of the each sequence that we are selected. From the full genome, you can get might be 100, 200, 150, and sometime a duplicate sequence is also available. So you can remove this duplicate sequence and can get the information. I mean, we can use only one gene. We have different genes that have the same information, so it's called duplication. So we remove the duplications and we determine the conserved motif structures. And next step is phylogenetic analysis of these genes that you are using. So for that purpose, the protein sequence should be aligned at the software, mega software. After that, we will determine or construct the phylogenetic tree based on the sequence that we are retrieved for each family. After that, the chromosome mapping. So phytosome database will use during when we download the file for the genome of a specific crop or specific species. So over there we have the chromosome position of each gene or the protein family that we are extracted for against specific condition. So over there we can collect all the chromosomal position of each gene and select its minimum and maximum value. So after that we use the map chart software. It's also an online freely available software. So we can construct the chromosomal mapping position on the gene on the of specific genes, I mean the map, it's a transcription family. After that, the R genes, uh, any type of gene that you are using and used to determine the genome-wide analysis for that. The first, next step is the antron exon distribution. For that purpose, we have the CDs data and the protein data, gene sequence data. It's combined used and draw the antron exon distribution using the gene structure display server. So genomic DNA and CDS sequence of all genes will be used to build the genome structure map for the antron phases. So basically, conserved domain analysis, I think I already discussed in the previous files. So uh, we can use the CDDD structure, NCBI's platform, and this final visualization we can check with the TB tool software. So the next step is the Salinity analysis. Salinity analysis basically is the comparison of the genome with the reference genome. So here I will clear you. If you are using for the legumes have a specific model plant like Medicago, and for the cereals, we are using about the Arabidopsis, the rice. So we can also making 2 or 3 genomes cumulatively check against the specific protein families, gene family or transcriptome family uh, uh, of the selected crops. So sanity analysis will provide the information of the 2 or 3 comparison genomes of specific gene family. So every software I provide the link in the PowerPoint. So if you need any help regarding the software, so it's available online, all the softwares except the TV tools. When we finish all the analysis, I mean the chromosomal mapping, intron, exon distribution, conserved domain analysis, the phylogenetic analysis, and then the centenary analysis, we select some genes from these specific conditions that we are using. I mean, here we are talking about the abiotic stress. On the abiotic stress, the salinity. So we have a number of genes that comes from the library, transcriptome library from the from the salinity experiment, and we use these genes and check the expression level. So it will give us the clear picture that these specific genes are really helpful and can be used uh, in the future to express, because these are the genes are the salt tolerant genes available at the end. So for that purpose, we are using, at the end we construct the heat map expression using this fold chain difference is either collected from the root or from the leaf to validate the study. So the things that I am discussed in the previous files, so this is the detailed information you can get if you go for the analysis. So you will get the gene ID, locus ID, chromosomal number position of these genes. And this is the start position, this is the end position of the gene. So this is the size, this is the protein accession numbers, and when a protein number. So this type of data you will get at the end. This is the chromosomal mapping conditions that you can make after the experiment. So this is the conserved domain analysis and antron exon distribution of a specific family. Suppose this is the green one, it's your specific family. So all the genes that we are selected have the same genome or conserved domain for a specific variety for a specific protein or the genes. So if somewhere it is not present, so you can remove these genes or the protein sequence from the analysis because they don't have the specific conserved domain in the analysis. So this is a type of phylogenetic tree that we can construct from the sequence that we are selected. So this is the heat map. Basically, here the four type of exposures time used by the researchers, and after that the expression profiling provide the information of a specific genes upregulated or downregulated under the control and the stress condition. Basically, it is the validation of the studies. So thank you very much.